So our next speaker is Kevin LaBar, who's a professor in the Center for Cognitive Neuroscience at Duke, as well as the Department of Psychology and Neuroscience. And Kevin has been a leader of the last 15 years or so in studying the interaction between emotion and cognition with an emphasis on memory. He's the recipient of numerous awards, including Young Investigator Awards from the National Alliance for Research on Schizophrenia and Depression, the Cognitive Neuroscience Society. He's a recently elected fellow to the American Psychological Science Society, and it's a pleasure to have Kevin here.
Okay, thanks very much to the organizers for inviting me to be here. I'm going to be focusing on emotional memories and looking at brain imaging techniques to look at the brain regions that might be specialized for encoding emotional information into memory and retrieving it from memory. And I'm going to be focusing today on looking at enhancing effects of emotion on memory. I'm not going to have too much time to talk about the impairing effects of emotion on memory, but maybe we can talk about that in the discussion section. And I'm going to be focusing on the form of memory that Anthony introduced, that is declarative or episodic forms of memory. And I'm going to start with how we study this using basic laboratory paradigms where we present people pictures and words that vary in emotional content. I'm going to be looking at emotion effects across both the encoding of information as well as at the retrieval stage. And then I'm also going to consider what happens when we try to regulate our emotions. How does that affect the brain regions that encode information into long-term storage? And then I'm going to be moving more from these basic laboratory paradigms into what we call kind of real-world situations, where we're going to be looking at memories for more complex social events, like a complex sporting event, and then finally what happens as we're retrieving emotional autobiographical memories from our personal past. Now, to start out with, this idea of marrying emotion and memory is very complicated, and so there are various theories of memory, there's various theories of emotion, and we have to kind of choose where we're going to look at that intersection. And today I'm going to be focusing on what's called a dimensional theory, which basically argues that emotions are organized in a space that spans two dimensions: the dimension of arousal, or the intensity of the emotional experience, versus pleasure, or the valence dimension, that is how positive or negative that information is. And so we're going to be systematically manipulating arousal and valence and then looking at the effects on memory. I'm not going to talk about these other theories today. And then in terms of memory systems, we also have to choose what system of memory we're going to look at. So Anthony introduced this earlier today in terms of this taxonomy of memory. According to this sort of approach, there's one brain region that plays a particular role in emotional remembering, and that's the amygdala. The amygdala I have highlighted here. It sits in the medial temporal lobe right in front of the hippocampus that Anthony introduced. So if you take a pencil and you stick it through your ears, you will eventually touch the amygdala. That has been implicated in the fear conditioning literature that we heard about from the last speaker. I'm not going to provide evidence for that today, although I can, again, talk about that later. Instead, I'm going to be focusing on this other role of the amygdala in memory that's been advanced by Jim McGall, who's going to talk about it after me. This idea that the amygdala will modulate processing in the hippocampus and other brain regions to enhance declarative forms of memory. And I'm going to be focusing on how we can look at this using brain imaging tools. The way that we do this, using this sort of basic laboratory paradigm, is to present people with pictures that systematically vary in their arousal and valence properties. We have them encode them while their brain is being scanned using functional magnetic resonance imaging. I don't have too much time to introduce that technique, but I just want to say that what this technique does is it looks at local changes in blood oxygenation and blood flow that's supplying the oxygen to the local neurons, so we can get that are participating in your tasks, so we can look at changes that are task-related in localized parts of the brain. And we're going to be scanning people while they encode these images. And then outside the scanning environment, we're going to test their memory for them. And what we want to do is to try to identify brain regions during the initial encoding that support good memory later on. And what we find on our memory portion of the study is that both pleasant and unpleasant pictures are remembered better than neutral pictures. Okay, so what this suggests is that the main driving force of emotion on memory is the arousal, or the intensity dimension of the emotion. The valence doesn't matter as much, at least in terms of overall percent correct here. And when we look in the brain at the time of encoding, we see that this part of the brain, the amygdala, is more active to both the pleasant and the unpleasant stimuli relative to the neutral pictures. So again, the amygdala's profile during encoding also matches the emotional intensity of the experience, doesn't care so much about the valence of that image. But the question is, how do we then relate this activity at encoding to what people remember later on? Is there a link between the amount of amygdala activity at encoding to what is remembered later? So to introduce this, we use a technique called the subsequent memory paradigm that Anthony and others have used here, where for each individual subject, we can identify the specific pictures that they subsequently remembered and those that they subsequently forgot. And then we can compare the brain activity for those items, pooling them together for each individual subject, and do a subtraction or a difference score. And what that yields is it's a signature of the brain regions that give you successful encoding operations that lead to good memory later on, okay? And then we can compare that for the emotionally arousing pictures versus those that are emotionally neutral. And this is just a 3D reconstruction of the medial temporal lobe of one subject, showing you the amygdala in red, the hippocampus in blue, and the surrounding cortices. And when we do this subsequent memory procedure, what we see is that in a lot of these structures, the amygdala and the hippocampus and the surrounding cortex, they show stronger activity at encoding that predicts subsequent memory for the emotionally arousing material than for the neutral material. But more interestingly, in support of this idea that the amygdala is modulating memory processes in the hippocampus and the adjacent cortex, we see greater correlated activity among these structures in individual subjects. So that is, if your amygdala was really hot when you were encoding the emotionally arousing item, your hippocampus would also be hot, or your entorhinal cortex would also be hot. And so what that means is that we have a very stronger functional coupling among these structures during encoding for emotionally arousing information, and not so much, in fact, not at all, in terms of the encoding of neutral information. So there is something special that's going on at encoding in terms of linking these emotional representations in the amygdala with the processes in the hippocampus to help bind that information into long-term memory. Now, we've just finished a meta-analysis of all the studies in the literature that have used this procedure. And indeed, what you find is that the amygdala and the hippocampus are the most consistently activated in these studies. There are a few other regions that also show up. So parts of the visual cortex, most of the studies use pictures or words. And so visual processing will also contribute. Attentional processing will also contribute. This is a region in the parietal lobe that's important for attention. And also regions in the frontal lobe that support deep semantic encoding will also predict memory preferentially for these emotional stimuli, okay? So it's a distributed network, but clearly the amygdala and hippocampus play an important part. Okay, the next question that we then turn to is to think about, okay, well, is this an obligatory circuit? That is, just if you present something emotional in the environment, does the amygdala automatically process this and lead to this memory benefit? But it turns out that it depends on what the individual is doing when they're confronted with the emotional material. So people will regulate their emotions in different ways, and this is work following James Gross here at Stanford. And so we wanted to look at how people's coping strategies or emotion regulation strategies might modify this relationship. And so to do this, we studied two forms of emotion regulation, one called reappraisal, where you basically cognitively change the meaning of the stimulus as it's being encoded in order to dampen its emotional impact. It's the basis of cognitive behavioral therapy. You can do it in several ways. In this particular experiment, we had people try to personally distance themselves from the emotional picture, make it not personally relevant. You could also do it where you try to imagine a positive outcome that happens as a result of this negative experience. And that has been shown to be very beneficial in terms of emotion regulation. And another strategy that's not so beneficial is called suppression, where you actively try to inhibit any outward expression of emotion to the stimulus. Okay? So we're going to compare what happens in these brain regions when you're reappraising or suppressing these emotional pictures. And what you see when you just get valence ratings to the stimuli, and this is on a scale where the most negative is at the bottom and neutral is 3, that for when you're just passively viewing the stimulus, you have a very low valence rating, so it's very negative. And you can actually, through both reappraisal or suppression, you can attenuate or alleviate the negativity in the stimulus. It doesn't get totally neutral, okay? So this is not looking after years of cognitive behavioral therapy. This is just trial by trial. Once you get one shot at this for each image. So you can attenuate the negative impact of the picture. But what's interesting is if you compare memory performance under these conditions, you have actually beneficial memory under the reappraisal condition relative to the suppressed condition or the other conditions. And so when you compare those effects in the brain, what you see when people are reappraising the stimulus is you get this nice tight coupling between the amygdala and the hippocampus that supports subsequent remembering, as I showed you in the past slide. You also get it in this region of the frontal lobe as well. So the frontal lobe is also communicating with the hippocampus under this circumstance. But in the suppression case, that's not happening. Okay? So the amygdala and the ventral prefrontal cortex is disconnected with the hippocampus. There's no relationship to support subsequent remembering. And so even though we might have people experiencing the same emotional event, how they process that emotional event can have implications for their subsequent memory. Okay, so far we've talked about processes at encoding that lead to the formation of emotional memories. What about during retrieval? So do these brain regions also contribute at the time that you are retrieving an emotional event from memory? So to do this, we brought subjects back from our first study and simply showed them the pictures that they had before, and this was a year later, so it's a year-old memories, and combined them with new pictures that were not in the original set, and simply asked them were these pictures in the original set or not. And we could do the same kind of analysis where we compare items that they successfully remember versus those that they don't, and do the same kind of subtraction. So you can consider this a successful retrieval index. And then furthermore, when we retrieve, we can use different levels of processing, and you'll hear more about this later, I believe. Sometimes when we retrieve, we have contextual details that come to mind, and this is in the literature, in psychological literature, referred to as a recollection, where you recollect some details about that image or the context in which you experienced that image before. Versus familiarity. You just know that this stimulus was on the list. It doesn't really come with any contextual details. And it's been shown that when we retrieve emotional events, it specifically affects recollection processes and not familiarity processes. So we want to look at that in the brain as well. And when we do this, again, we see our similar players here, the amygdala, the hippocampus, and the entorhinal cortex, will show more activity for successfully retrieved information relative to unsuccessfully retrieved information. And it does that more for these emotionally arousing pictures than for the neutral pictures. And again, if we look at the connectivity among these regions, they're more directly connected for the emotional retrieval than during the retrieval of neutral material. And then when we break this down into items that they remembered with contextual detail versus those that they said just seemed familiar to them, we see this boost in both the amygdala and the hippocampus at the time of retrieval that's correlated with this subjective sense of recollection, okay? And we don't see this in other brain regions, so this is just a neighboring region in the medial temporal lobe that responds during retrieval of emotional memories, but doesn't make this distinction between recollection and familiarity. So we think that this boost in the amygdala and hippocampus not only helps you retrieve the information, but it does so because it provides some extra contextual, detailed information. Okay, so how then can we move from these basic paradigms to looking at more complex, real-world emotional memories? You know, looking at these pictures or words that we present in the lab, does this have anything to do with the more complex kinds of emotional memories we have in the real world? And so there's been a call in psychology. Paul Rosen wrote this review that basically says there should be more interest in what people actually do: eat, have political views, watch television, et cetera. and more concern about whether the instances we choose for our psychology experiments correspond to real-world events and are both robust and generalizable. So I'm going to spend the next half of my talk extending from these basic paradigms to more complex forms of memory. And what do people do at this time of year? They watch basketball, okay? So we are in the middle of March Madness, even though it's not March anymore. And so people are watching basketball games, and we thought that it might be interesting to use a basketball sports competition paradigm to look at the encoding and retrieval of emotional information from memory. People get pretty heated as they're watching these games. The games are pretty intense. It actually provides a very nice paradigm to look at moment-by-moment changes in emotional valence and intensity and how that affects memory. And I'm also at Duke. So Duke and UNC are very competitive. Duke won the national championship last year. UNC won it the year before. This year, neither of them will win it. But anyway, so we have this great fan base in the Triangle area. So we had them watch an archived game. This is sometimes called a stage events paradigm in psychology, where you have everybody come in, you watch an archived game. We did it in small groups on a big screen, and the groups were rooting for the same team. What's interesting about this is that the valence is determined by the perspective of the fan, whether the particular shots of the game were beneficial to your team or beneficial for the other team, right? So we don't have to present separate stimuli that are positive and negative. The same event will be coded as positive or negative depending on your perspective. What we assessed in our memory was specific plays of the game, specific shots of the game. What's really nice about this is it allows us, basketball in particular, there's lots of shots of the game, and whether the shot goes in or not determines the emotional significance of that particular shot. And so the shots of the game provide kind of narrative points where each point has a different emotional impact. And so we can kind of track this with imaging. So what we did was we brought people back after they watched this game, and we showed them clips of the game where a player took a ball from one side of the court to the other, and we ended the video clip right at the point where the shot was released towards the basket. And your memory task was, did this shot go in or not? All right, so that's your memory task. And then we can have them code the emotional impact of that event. All right, and so I'm going to be looking at amygdala and hippocampal activity as a function of how emotionally intense that memory was rated by the participants. And what we see, again, in both of these brain regions is that these regions are encoding the emotional intensity of that shot. So some shots are highlight reel plays. They're really exciting. It didn't matter whether you were a UNC fan or a Duke fan. As long as you rated that thing as emotionally intense, you got more activation in the amygdala and hippocampus while you're retrieving that shot. And we also see that emotional intensity has a broader reach over other brain regions that we don't see in our regular laboratory studies. So for example, we see changes in midline frontal regions of the brain and posterior regions that are thought to be important for self-referential processing and social cognition. And we see that those brain regions are also moderated by emotional intensity. And these brain regions we don't normally see when we just present people with pictures or words, okay? Now what about the valence effect? People tended to remember better the shots of the game that benefited their team than those that benefited the other team. And this positivity bias in memory was linked to brain regions that are implicated in attentional processing, the frontal lobes and the parietal lobes. And this goes with other social psychology literature in terms of attentional broadening under positive affect. So this is the only valence-specific effects that we saw. Again, most of the effects were driven by the emotional intensity of the shots of the game. Okay, so finally, I want to talk about the most unconstrained situation where we just bring people in and have them retrieve an autobiographical memory from their personal past, okay? So in order to do this, we use an old technique in psychology where we simply present a keyword, like the word picnic, and we scan the brain as you try to choose from the many picnics that you've experienced in your life, a single picnic event, and we have people indicate with a button press when they have a specific event in mind, and then we have them hold that memory in mind and try to relive that memory for a period of time until the end of the trial. And then at the end of the trial, they encode how emotionally intense was that memory and how much did they relive or re-experience that memory. And what's nice about this paradigm is that it takes people quite a while to generate a full-blown memory from this generic cue. And so we can actually look at this reconstructive process of memory as we talked about, as we heard in the earlier talks. And so we divide up our trials to the time in which they're retrieving and selecting a memory and reconstructing the memory. And then we can also look at the time where they're just maintaining the memory in mind until the end of the trial. Okay? I'm just going to show you a small set of data here, but when we look in the amygdala and in the hippocampus, what we find is that those brain regions, again, correlate with the emotional intensity ratings. And very interestingly, they show up early on in the act of retrieval. That is, as the memory is being assembled and constructed, those brain regions are more active, and they correlate with the later emotional intensity ratings that are given. Whereas if we look back in sensory processing regions like the visual cortex, we see more activity later in the trial when you're just holding the memory in mind, and that activity correlates more with the reliving ratings than the emotional intensity ratings. So here we can actually use brain imaging to try to tease out the dynamics of what's going on during the act of retrieval. And what my colleague David Rubin likes to say about this amygdala result is that the amygdala knows you're about to have an emotional memory before you do, right? Because the memory is not fully constructed at this time, but there's this early signal in the amygdala and the hippocampus that's telling you something emotional is about to come online. And so this goes with the older literature in psychology regarding the reconstructive aspect of memory by Bartlett, where Bartlett said when a subject is being asked to remember, very often the first thing that emerges is something of the nature of an attitude. The recall is then a construction made largely on the basis of this attitude, and its general effect is that of a justification of the attitude. And what Bartlett was talking about in attitude was largely a matter of feeling or affect. So the feeling or the affect comes on first, and then the details kind of get filled in. And we think that we can now use brain imaging to start looking at this process as it unfolds. Okay, so to conclude, I want to say that emotional memory is a multidimensional construct. There's not a single effect of emotion on memory. And I've only given you a small taste of that today. That the amygdala and adjacent medial temporal lobe regions are important for the long-term encoding and retrieval of emotionally intense episodes, and you get some additional support from frontal parietal and some sensory regions. That how you regulate your emotions in the moment will modify these interactions and can affect the representation of those events. And that these interactions extend to more complex social memories and memories from our personal past. And when they do so, emotion has a broader reach over regions, for example, those that are involved in social cognition. Okay? And then I finally want to just conclude by saying where we're going from here. And we heard in the last speaker in terms of the role of spatial context and how contexts are important for reinstating memories. And at Duke, we have a technology called the Duke Immersive Virtual Environment, where you are completely surrounded by computer projection screens, and you have virtual reality goggles on, and we can actually immerse you fully into scenarios and present objects as you navigate through these scenarios, and we can then test your memory for these objects and look at what happens when you're presented those objects in the same context that you encoded them, or what happens when you have a context shift. And we think this is going to be very important for understanding things like PTSD, where they tend to have memories for triggers, emotional triggers, outside of their initial encoding context. So it gets us a way to look at how you can use virtual reality technology to look at the contextual control or retrieval of emotional memories. So I just want to thank all my collaborators on this. There's lots of people that have helped out, NSF and NIH for the funding, and the students in my lab. Thanks very much.
2 questions, my handlers tell me.
I have just an odd little technical question. I noticed when you went from some of your earlier studies to the ones with the basketball, that
Yes.
the amplitude of the bold signal went up by a factor of something like 10 or more. I was wondering if you thought that was meaningful, that the scale of the signals.
I haven't actually directly compared the scales, but you might be right, that once you move into these real-world contexts and you have these very strong emotion manipulations, that the signal change is probably a lot stronger. And that does raise another issue that's important, I think, maybe even for the general discussion, which is that it's very hard in laboratory to manipulate the emotional intensity ethically in a way that you might experience in the real world. And so using something like this basketball paradigm might allow you to get at that.
I was just wondering, in your final experiment, the autobiographical one, when you were eliciting memories using these words, whether you factored in the age of the memory, how long ago these things, because that would be interesting theoretically and also for legal consequences.
Yes, and the problem, well, we have, so we ask people, what is the age of the memory? And the issue is that most people will give you memories that are fairly recent in this particular context. So most of the memories are coming from the last 5 years, generally speaking, because those are the most accessible usually when you give a generic cue like this. Although you can see something called the reminiscence bump, where you tend to remember things from like your 20s and 30s if you're old enough. These subjects were not old enough to show that pattern, okay?